All right, so good afternoon, everyone. My name is Brodie and I'm a PhD student at the University of Adelaide. Now, before I start, I'd just like to say a massive thanks to ABSS for allowing me to speak at this conference, and to my supervisory team, Associate Professor John Semler, Dr. George Opie, and Dr. Simran Sidhu for their invaluable guidance. So today I'll be discussing the influence of regular physical activity on neuroplasticity following a bout of acute aerobic exercise as assessed with transcranial magnetic stimulation, or TMS. So given it's been discussed so thoroughly, I won't really get into the mechanisms of TMS, but very briefly, it's a form of non-invasive brain stimulation which we've used and is commonly used over the primary motor cortex to generate motor evoked potentials, or MEPs, in peripheral muscles. The amplitude of these MEPs is used as an index of corticospinal excitability, and a change in mean MEP amplitude is thought to indicate the induction of plasticity. It's worth noting, and as is relevant to this study, in addition to its use for the measurement of excitability, TMS can be used as a plasticity-inducing paradigm, and accordingly, numerous studies, including these two, have used TMS as both a measurement and interventional tool to investigate the influence of regular physical activity or physical activity on brain function. So this graph on the left, which was also described by Ashley Smith earlier on, is adapted from a study which investigated the influence of regular physical activity on plasticity. This study used a plasticity-inducing TMS intervention known as paired associative stimulation, or PAS, which is known to upregulate corticospinal excitability. Data represent MEP amplitudes recorded before the PAS intervention, as depicted by the blue columns, and after the PAS intervention, as depicted by the yellow and pink columns. We can see that there is a significant upregulation of MEP amplitudes after the PAS intervention in the active group, whereas there is no change in the sedentary group. The graph on the right-hand side has been adapted from a recent study which investigated the influence of high-intensity interval training, or HIIT exercise, as I'll refer to it, on neuroplasticity using an alternative plasticity-inducing TMS intervention known as intermittent feeder burst stimulation, or ITBS, which is also known to have a facilitatory effect on corticospinal excitability. Data represent the mean MEP amplitudes recorded after ITBS that have been normalized to the pre-intervention time point. And we can see here that normalized MEP amplitudes are greater following ITBS when preceded by HIIT exercise, as illustrated by the pink squares, compared with the rest condition, whereby ITBS was preceded by a no exercise control, as depicted by the blue circles. Together, these studies indicate that both acute and regular forms of exercise influence neuroplasticity. However, it remains unknown how regular physical activity influences neuroplasticity following an acute bout of exercise. So accordingly, the aim of this study was to investigate if cortico-motor plasticity following acute aerobic exercise was modified within individuals with a history of endurance training, and it was hypothesized that a history of physical activity would promote greater cortico-motor plasticity following acute exercise. The study was a crossover design that included 2 experimental sessions that were randomized in order. One session included an acute bout of HIIT exercise, and another was a control session. 28 subjects took part in this study. One group consisted of 14 endurance-trained cyclists, ranging from highly motivated recreational riders and local racers right through to professional tour-level cyclists. And a second group consisted of 14 sedentary individuals who did not complete any structured exercise regime. EMG recordings were taken from the first dorsal interosseous muscle of the right hand. As we can see on this protocol diagram, we began with baseline TMS measures of corticospinal excitability before participants completed either the HIIT exercise block or an exercise control block. The HIIT exercise block included 4 separate blocks of 2 minutes at high intensity, interspersed with 4 3-minute blocks of stationary cycling at a low moderate intensity for a total duration of 20 minutes. For the exercise control session, participants sat quietly on a bike without pedaling for the 20-minute duration. 10 minutes after the completion of exercise, TMS measures of corticospinal excitability were reassessed, and 25 minutes after exercise completion, a plasticity-inducing TMS paradigm known as ITMS was applied. This intervention involved 180 pairs of TMS stimuli lasting approximately 15 minutes and is also known to have a facilitatory effect on corticospinal excitability. TMS measures of corticospinal excitability were then reassessed both 5 and 30 minutes after the ITMS intervention. Okay, so now we move on to the outcomes, and this graph displays the influence of HIIT exercise or the no exercise control on corticospinal excitability. The data represent the mean amplitude of MEPs from after exercise normalized to baseline, with values over 100 indicating greater MEP amplitudes after the exercise. We can see that MEP amplitudes were not differentially modulated between groups after the HIIT exercise. However, surprisingly, the endurance train group displayed greater normalized MEP amplitudes after the no exercise control block. The reason for this is unknown, and it's difficult to determine with the present data. Now we move on to looking at the effect of ITMS on corticospinal excitability when preceded by either HIIT exercise, as is displayed on the left of this graph, or the no exercise control, as is displayed on the right-hand side of this graph. Data are pulled across 5 and 30-minute post-ITMS time points and are normalized to the post-exercise or pre-ITMS time point. The primary and novel finding of this study is in line with the hypothesis and is shown on the left-hand side of this figure. We can see that when preceded with HIIT exercise, ITMS-induced modulation of MEP amplitude is greater in the endurance-trained cyclists compared with the sedentary group. This provides evidence that regular physical activity promotes the upregulation of plasticity following an acute bout of exercise. Furthermore, this study supports recent work that suggests that acute exercise in itself promotes the response to experimentally induced plasticity, as we see greater modulation of MEP amplitudes following ITMS in the exercise session compared with the control session. So to summarize, this study supports existing work that suggests that acute exercise promotes TMS-induced plasticity and provides novel data to suggest that regular physical activity enhances the effect of an acute bout of exercise on facilitating plasticity. Further research is required to determine what particular characteristics of regular physical activity, such as time, intensity, and modality, contribute to the facilitation of acute exercise-induced plasticity. Thank you all for listening. I now invite any questions.
Very much, Brodie. We do have one already. Brodie, great talk. Oh, sorry, it's from Kate Hoy again. Great talk. Was there any difference within the endurance trained group in light of their range of experience or ability? It's a good question. I had that same one myself.
Not really. So there's no correlations. I assume you mean between the bottom end of the recreational cyclists and the professional sort of level cyclists.
I suspect so.
Yeah, no, there's no real correlations present. There was 2 data points or 2 subjects which sort of, they weren't statistically outliers, but they were definitely further away from the mean than others, and both of those individuals sat about right in the middle of the data pool, to be honest. So no, not really. I will mention though that when I say highly motivated recreational riders, we're still talking guys that are cycling for 10 to 15 hours a week. So all of that endurance trained group were fairly trained.
Right. Excellent, thank you. I had a simple question about the choice of intervention. So you have any particular reason you chose the ITMS versus some other plasticity inducing protocol? Is something interesting about that protocol that you thought would be good for this particular design?
Not anything other than the fact that evidence suggests that ITMS potentially has a bit more reliability or a little bit lower variability than some other plasticity-inducing TMS measures, and we've used it fairly extensively within our lab group in the last few years, and we've got data which both is published and is still yet to be published, and we have found that it's a little bit more consistent in inducing an LTP-like effect than, say, ITBS, for example. So that was the reason for picking ITMS in this case.
Great. We do have another one come through now. So it's from Nigel Rogash. So thanks, Brodie. Was the HIT protocol a cycling protocol? And if so, would you, or if not, would you expect a similar result if you used an exercise paradigm which one group were not highly trained in, for example, cyclists doing running or something like that?
Yeah, it's a really good question. So to answer the first point, yes, the HIT exercise protocol was a stationary cycling protocol, and it's a little bit hard to identify whether we would see the same things if it wasn't, I suppose, a protocol which is familiar to the participant group. Given that there's the potential that some of this modulation is a result of sort of chemical and cerebrovascular factors, it would be expected that I would expect we would see similar modulation through any endurance or aerobic style exercise. However, based on some studies by other groups, you probably wouldn't expect to see the same thing if it wasn't an aerobic form of exercise. So for example, if it was a strength training task, I would say you're less likely to see modulation like we did.